And so welcome everyone. This session is called Ecosystem Approaches to Fisheries and Management. We're happy to have you participating in virtual homecoming. I hope you're enjoying yourselves and learning a lot. And I hope you have the opportunity to connect with people. My name is Anne-Marie Reddy and I'm the host of this session. I'm actually co-hosting with Tia Bullard, who is the T Bullard that you see on the screen. And I'm very pleased to introduce Dr. Gavin Fay. He's the Associate Professor of Fisheries and Oceanography in the School for Marine Science and Technology, lovingly referred to as SMAST. Dr. Fay's work focuses on using statistical and mathematical models for better ecosystem-based decision-making for fisheries and our oceans. He teaches courses in statistics, ecological population modeling, science communication, and ecosystem-based fishery management. That was a mouthful. He currently serves on the Mid-Atlantic Fishery Management Council Scientific and Statistical Committee, as well as in the New England Fishery Management Council's Ecosystem-Based Fisheries Management Plan Development Team. His work has brought him from the Northeast to Alaska, to the West Coast, to Europe and Australia. Dr. Fay is joined by some of his students whom he will introduce, but before letting him do so, I want to let you know that there will be time at the end of the presentation to ask questions. And I think probably the best way to do that is to either type in the chat that you have a question, or you can type in your question, or if your camera is on, you can just simply raise your hand and I will make sure that we get to you. So thank you very much. And again, Dr. Fay, thank you and welcome.
Thank you very much, Anne-Marie. It's really nice to be here. Thanks everyone for joining us. I am going to go ahead and share my screen. So just bear with me while I do that. And can everybody see the slideshow that I have here?
Yes.
Fantastic. All right. Well, I say thank you everyone for joining us. My name is Gavin Fay. I'm an associate professor at the School for Marine Science, Technology, and you're going to hear from me for a little bit right now, but not very much because I'm going to let the voices of our students tell you about the work that is happening at SMAST. Okay, there we go. All right, so our oceans today are impacted by a multitude of challenges and opportunities. There's a lot of uses and services that we derive from our oceans. And when we think about humans managing our oceans, we're thinking a little bit about fisheries, but also a larger context within which healthy oceans exist, including humans interacting with fishing, energy use, shipping, tourism, land management, and land reclamation from the sea. So our fisheries and seafood that we derive from our oceans really sit within this large system. And increasingly, there's been this push for something called ecosystem-based fisheries management, which I agree, Anne-Marie, is a little bit of a mouthful. Really, EBFM, or eco, is the buzzword that is used, is really about the systems part. So I like to think, if I can forget about the eco and the based part, and I go systems-based fisheries management, it's really this recognition that our sort of fisheries are existing within this larger system context. And so there's, how can we make decisions for our fisheries and other activities in the oceans, recognizing that there's these coupled systems of ecological, environmental, social, cultural, economic, and governance relationships all through these marine and coastal systems. Because we have, our fisheries sort of are deriving food production, but they're also providing jobs and the source of livelihoods for coastal communities. And so thinking about managing activities in the oceans, we're really thinking about trade-offs and balancing between sort of healthy ecological and healthy ecosystems, healthy fish populations, but also thriving coastal communities with that have food security too. So this really means that we need a workforce that is able to think interdisciplinary and is also able to collaborate amongst these different parts of the system in terms of bringing expertise to bear, whether that's ecologists, economists, policy makers, spatial planners, social scientists, and the industry themselves. And so we're really fortunate at UMass Dartmouth to have the School for Marine Science and Technology, or SMAST, which is really focused on this interdisciplinary approach to the marine sciences. We have graduate programs, master's programs, PhD programs, and professional science masters in all a multitude of disciplines and fields within the marine sciences. And a big strength of our program, I think, is that we emphasize collaboration and knowledge co-creation with regional industry, with government, and also non-governmental agencies to improve kind of our systems understanding for marine environments and supporting sustainable fisheries. And we all know, living close to New Bedford, we all know how important fisheries are to the south coast of Massachusetts. I should say we also, in addition to graduate training, we also have lots of undergraduate research opportunities, including undergraduate technician positions and internships as well, including a new research internship course that will be happening in the spring of 2021 for upper level undergraduates. So please get in touch if you're interested in that. Okay, so I'm now going to sort of give you an overview of the structure for the remainder of the session. We're going to have two sort of sub-sessions within the session. We have 7 students here today, both master's and PhD students, who are going to give us 8 talks. We're going to separate those into 2 sets of 4, so we don't have a massive sort of fire hose dump of information all at once, and give everybody a chance to ask questions after each of those sets of 4 talks. The first session is going to be loosely around research focused on ecosystem approaches to sustainable fisheries. And for that, we have Alana Minich, Kim Sawicki, Tricia Perez, and Amanda Hart. And as Anne-Marie said, we're going to give time for questions after these talks. So if you have questions during the talk for our students, just go ahead and either write those questions down in the chat, or once we get to the end, raise your hand and we can call on you and you can learn more. So without further ado, I am going to hand over to our first speaker, Alana Minich.
Hi, everyone. Thanks, Gavin, for the nice introduction. I'm Alana. I'm a PhD student here at UMass Dartmouth, and today I'm going to be talking about biologically driven management of the Atlantic bluefin tuna. So the Atlantic bluefin tuna is the largest tuna species. One fun fact about the Atlantic bluefin tuna is that unlike most other fish, they're actually warm-bodied, and their large size and the high swimming speeds contribute to their place as an apex predator. They're considered a trophy fish among hobby fishermen and also support a high-value fishery due to their strong demand in the sushi industry. The transatlantic crossings of Atlantic bluefin tuna are well documented, and they're capable of inhabiting a range of temperature regimes from subarctic to subtropic. Current management of this species assumes 2 stocks, an eastern stock that spawns in the Mediterranean Sea and a western stock that spawns in the Gulf of Mexico. However, a recent discovery of Dazel larvae in the Slope Sea calls into question the exclusivity of the Gulf of Mexico as the western stock spawning ground. So how can we determine the importance of spawning in the Slope Sea for the population of Atlantic bluefin tuna? We can do this using biogeochemistry. So isotopes of an element have the same number of protons, but a different number of neutrons. The heavy isotopes of an element naturally comprise a smaller percent of total abundance, so they're perfect to use as natural tags. Natural tagging relying on stable isotopes utilizes geographic differences in isotope ratios that are then recorded in the chemical composition of body tissues. To determine the potential contribution of the slope C to the Western stock, I'll be utilizing carbon and oxygen isotopes, which can tell us in what water masses a fish has spent time. We can identify geographic differences in oxygen and carbon that are naturally present in seawater to distinguish between larvae spawned in the Gulf of Mexico and those spawned in the slope sea, and then use those signatures to identify the birthplaces of adults. This is accomplished by analyzing the otoliths of the fish. Otoliths are calcium carbonate structures that are basically fish ear bones that develop over the course of the lifespan by pulling carbonates from seawater to form. In larvae, the entire otolith will be reflective of the spawning site because the fish has not spent time anywhere else. So this is our baseline signature. We can then compare these signatures to adults by testing the core of the larger otolith to see which place of origin their chemical markers match to. This will serve as a preliminary test of the importance of the slope sea as a spawning ground to the western stock of Atlantic bluefin tuna. This is relevant for the management of Atlantic bluefin tuna and their sustainability as a fishery because both the eastern and western stocks have suffered overfishing in the past. The eastern stock has made a remarkable rebound into a recovered biomass, but the western stock hasn't seen as much recovery. If we identify the slope sea as a significant spawning site, conservation measures can be taken to protect adults utilizing the slope sea during spawning. We can also develop more accurate models, which can help predict changes to biomass. To ensure sustainability of the Atlantic bluefin tuna fishery, the role of the slope sea in western stock biomass must be understood now and moving forward in a changing ocean landscape. Thank you.
Great, thank you, Alana. Please, everybody, use the emoji button at the bottom of your screen to thank Alana for her talk. One of our students is having some trouble getting into the Zoom, so I am actually going to skip over the next talk to allow them to get in. And if Trisha, if you don't mind, it'd be great if you could give your talk.
Sure.
Oh, I went too far, sorry. There you go.
All right.
Go ahead. So our next speaker is Trisha Perez.
All right. Yeah, so my name is Trisha Perez. I'm a master's student at SMAST in the Stokesbury lab, and my research is on George's Bank Yellowtail Flounder. George's Bank is a large raised bank about 65 miles offshore from Cape Cod and was once extremely productive with groundfish species such as cod, halibut, and yellowtail flounder. And this fishing ground helped to build up the fishing communities and ports all around New England. The George's Bank Yellowtail fishery that once caught over 20,000 metric tons, about 44 million pounds a year in the 1960s, only landed 8 metric tons in 2019. And despite lowered quotas set by management, this quota, the allowable catch limit, has not been reached by the fishery fleets in the last decade. Stock assessment shows that abundance and biomass continues to decrease as well. And on top of that, federal surveys are showing an aggregation of yellowtail flounder, the orange circle on the bottom left graph. So, and this aggregation is in closed area 2, which is a large marine protected area whose access by fishermen is highly regulated. Federal survey design limits how focused we can be on this aggregation of fish to try and figure out the true status of the population. But my lab at SMAST may have a solution. Since 2013, we've been seasonally assessing closed area 2 with the specific objectives of data collection on vulnerable flatfishes, including yellowtail flounder and window-paned flounder. Our video trawl survey has an open cod end, meaning we put a camera into the net and record the fish passing through that we can count later on land. But occasionally we do close the net so that we can do biological sampling on deck. My lab at SMAST also conducts a drop camera survey where we take photos of the seafloor to assess the scallop population, but this data set also has substrate data as well that can be used for habitat. And you can see the little yellowtail flounder, he's saying hi down there. And so I'm gonna use these 2 data sets to answer the question of why. Why won't the yellowtail flounder stock rebuild? Is it temperature change? Is it competition with other species? The protection that closed area 2 provides has resulted in the scallop fishery booming. And that's this third photo in the Y. Those are all scallops. So is the availability of habitat for yellowtail reduced by the increasing scallop populations? Or has the substrate, the habitat, changed over time from sand to rock or vice versa? And lastly, the scallop fishery is granted access to closed area 2 on a rotational basis. So are these periods of intense exploitation directly affecting the condition of yellowtail flounder? So my research is going to provide an additional estimate of the total amount of yellowtail flounder in this aggregation, which could educate the quota decision. And I'm acting as an ecological detective using my lab's available data set to hunt down answers as to why this once massive fishery has dwindled and will not rebound. Thank you.
Thank you, Tricia. Please show your appreciation for Tricia using the appropriate emoji. Thanks very much. Okay, so our next talk is going to be from PhD student Amanda Hart.
Gavin, I'm missing a picture on this slide.
Okie dokie.
Thank you. Awesome. Hi, everyone. My name is Amanda Hart, and I'm actually a PhD student in Gavin's lab at SMAST. And today I'd like to tell you a bycatch story, which begins with a single species of fish. Because when we think about how we're managing and regulating the catch in our fisheries, we only look at one species at a time. But when we actually go out fishing, we start to see that the story is a little bit more complex because there are multiple species of fish in our marine environment. And our fishing decks tend to look like this picture on the right, where hopefully we caught a lot of the fish we were trying to, but there's also a fair amount of bycatch occurring. And when we look at the management consequences of this, they're a pretty mixed bag. The species of fish we were targeting, this kind of pinky red fish, could still be healthy. We might have followed all of the regulations, the population is doing well, but there are these negative side effects that we really need to consider because our bycatch species might not be as healthy. So this, catching them in multiple fisheries could be a problem. And over time, the fisheries might start to suffer for this because in order to protect the bycatch species, we might start to restrict the catch of healthier populations like our redfish. And that has negative consequences for the fisheries that are trying to make money. So in my research, I'm trying to understand exactly where and how this bycatch is occurring and how it impacts our fisheries management. To do this, I'm looking at bycatch information in time and space. So I'm focused on Georges Bank, just off the coast of Massachusetts. And I'm looking to see where fishing is overlapping to help explain where this bycatch is likely to occur. And I'm also exploring environmental information because fish are hanging out in the environments that meet their needs. And as these environments change over time, where fishing occurs and where bycatch is happening is also likely to change. So that might be another useful source of information because as our environments are changing, we're also starting to change our management to ecosystem-based fisheries management. We're no longer looking one species of fish at a time, but we're considering multiple species, their interactions with each other, their environment, and the fisheries that catch them. And I'm trying to test some of these ecosystem-based fisheries management to make sure that at the end of the day, these management options have the highest amount of positive outcomes for the fish that we're trying to catch. And we are acknowledging but trying to minimize the negative outcomes from our fisheries. So bycatch is never going to go away, but if we're proactive and we design our ecosystem-based fishery management frameworks well, we might be able to minimize some of those negative consequences when bycatch does continue to occur. With that, I'll take any questions at the break, but please feel free to reach out to me in the meantime.
Great, thank you, Amanda. Everybody, please show your appreciation of Amanda's talk with the appropriate emoji. So unfortunately, it seems that Kim has been unable, still having some problems getting into the Zoom. So I am going to switch us to our question time. So if anybody has questions for Amanda, Trisha, or Alana, please go ahead and either sort of wave your hand right now or write your question into the chat, and we'll have a few minutes to answer those questions.
This is Anne Marie, and I have a question, and it's broad, but it made me think, you know, while you were talking about it, is most of the research that you're doing in the Atlantic coast in our area, or do some of you go further north or south to do your research?
My data set, it's Amanda Mealy. My data set ranges from Browns Bank, Canada to North Carolina for my project. So they do a little farther.
Yeah, okay. Thank you.
For my project with the bluefin tuna, I have larvae that have been collected from the Gulf of Mexico and from the slope sea around this area. And I'm hoping to, because they're such a migratory species, I'm hoping to evolve my sample set into involving adults that range across the entire Atlantic basin.
So when you do research that's at that great distance, how do you get your research? How do you get your data?
So for me personally, I used to work at NOAA, which is how we've collected our samples from the Gulf of Mexico. And so that involved going onto a ship and collecting the data. With the slope sea data here, that's through with a collaborator that I have also at NOAA, but the Northeast office. So he's sharing samples with me. And then for fish that I intend to obtain in the future as adults, those will a lot of times, I think the easiest way to get those without a time and research expense of going through a grant to go out sampling at sea will be to obtain those samples from fishermen who happen to be in the area of a certain range of distances.
Oh, fascinating. Thank you.
I had a question for Amanda. What environmental factors are you looking at on Georges Bank?
So there's a couple of different factors, but the major one is temperature, because we know that fish have thermal habitat preferences, which means that there's a range of water temperatures that don't cause any sort of physiological stress, and or that's where their prey likes to hang out. And so we're going to look at how the fish vary in space with temperature, and also like how fish are overlapping, like how the populations themselves are overlapping, which isn't necessarily a piece of catch information, but is heavily influenced by environmental information from temperature to bottom type, etc.
Cool.
All right, any further questions for three speakers that we've had so far? Okay, well, keep muddling on and thinking about them, and we will have time again at the end of the next set of talks to be able to come back to things that people think of them. And please feel free to write questions into the chat during the presentations, and our students can either answer them live or we can wait until the end too. Okay. Well, thanks very much, Amanda, Amanda, Patricia, and Alana. I'm going to transition now to our next mini topic here, thinking about, so that three talks before sort of talked about kind of thinking about ecosystem approaches of fisheries in terms of recognizing other effects other than fishing on our populations. And our next set of talks is really thinking about seafood production. So not just fishing, but also aquaculture, and recognizing that seafood production going forwards is dealing with several sort of changes in the system. So I will turn that over to our first speaker, which is Alison Fry.
All right, hi everyone. My name is Allie, and I'm currently working towards my master's degree with Dr. Steve Cadron here at SMAST. So today I'll be talking about Atlantic cod, which are one of the most well-known fishes in the waters of New England. Once so abundant, fishermen describe the oceans as being carpeted with cod. Many of you have probably heard of Cape Cod. Indeed, it was named after the fish. And dating back to the colonization of North America, humans have been fishing for cod as a valuable food resource. Upon the Industrial Revolution and development of highly efficient fishing gears, we began to remove too many fish, and the populations were not reproducing quickly enough to withstand these intense fishing pressures. In the later 1900s, the cod fishery collapsed, and since then, scientists have been trying to rebuild these once fruitful populations. In the waters off southern New England, cod appear to be behaving a little differently than their counterparts in the Gulf of Maine and Georges Bank, and little is known about them to say why. My team and I are conducting a study to assess the specific habitat preferences and dynamics of spawning and migration to help better understand this group of fish. To do so, we're using acoustic telemetry methods. This means that we head out offshore during the known winter spawning season, catch large reproductively mature cod, and insert acoustic transmitting tags inside of their abdomen. These tags release unique pings, which are received by 10 hydrophone buoys we have deployed around our study area. After downloading the data from these buoys, we can see which fish were where and for how long. The high spatio-temporal resolution of these data allow for us to draw conclusions about where these fish like to live, spawn, and also gives us a sense of their migration patterns. To make all of this even more complex, our study site falls directly within the offshore wind energy lease area. This area has been selected for the development and construction of over 100 wind turbines, which will offer New England a new renewable energy source in the near future. With all the excitement and promise of offshore wind energy development, it is still poorly understood how the construction and maintenance of this large wind farm will affect native fish species like the Atlantic cod. So the goal of our study is twofold. First, to broaden our knowledge of these southern New England cod and answer questions like, where do these fish live exactly? How long do they live here for? What is their preferred thermal range? And do they return to the same site year after year to spawn? This information will be valuable for management and setting fishing regulations. And secondly, our project aims to offer a baseline perspective of how these fish are using this area so that we can better understand the ecological impacts of offshore wind energy development. We're currently gearing up for the second winter spawning season of our study, where we will begin to answer many of these important questions. So for now, stay tuned, and you can follow along at my Twitter or email me. Thanks.
Thanks, Allie. Please show your appreciation for Allie with an appropriate emoji. Okay, so we're going to stay in wind farm world, and next talk is going to be from Amanda Melly, who's going to talk to us about Jonah crab.
Hi, I'm Amanda, a master's student from the Stokesbury lab at SMAST. The summary of data that I'm presenting today is part of our first year of our before-after control impact study for the Vineyard Wind Farm leasing area from 2019. This survey looks at targeted species before and after the wind farm construction. So our survey is focused on lobsters and Jonah crabs in the area. Lobsters were targeted since they are a current fishery in the southern New England area, and Jonah crabs were targeted because they are an up-and-coming fishery used to complement the lobster fishery. This survey was broken down into sampling periods, where each sampling period consisted of one setting and one hauling day, where catch was recorded and measured. We had a total of 8 sampling periods ranging from July to October. During that time, we were able to catch and measure a total of 1,889 Jonah crabs. The Jonah crabs range from 19 millimeters wide to 169 millimeters wide, where the majority of crabs were between 109 and 129 millimeters. The smallest male crab was 19 millimeters wide, and the largest male crab was 196. For females, the smallest was 46 millimeters, and the largest was 131 millimeters wide. About half of our Jonah crabs that were caught were either equal to or larger than the legal size minimum of 120 millimeters wide, which is shown by the red line in our length distribution figure. There were more crabs caught in later sampling periods, periods 7 and 8, than earlier sampling periods. This is interesting because it could show a fall migration where more crabs are traveling in or offshore from our area, so there were more caught in the traps. But we do need more information and more years of surveying to make a conclusion on that. We also found that only 4% of the Jonah crabs caught were female, which such a low percentage of females, it shows that maybe we missed their breeding period in that area, since our complementary larval survey did show an abundance of larval crabs in this area during the earlier sample periods. So it could be that this year, since we did start in May, we could see more females in that earlier sampling periods, which would make sense for our larval survey. And fun fact, the first Jonah crab stock assessment workshop will be scheduled for November of this year. So that will be very interesting and a start for this fishery. Thank you.
Thank you, Amanda. Please thank Amanda for a great talk. Our next presentation will be from Mike Coop.
All right. Hello, everyone. My name is Mike Kurt. I'm a master's student in Dr. Pingo He's lab at SMAST. And I'd like to talk to you today about aquaculture, switching a little bit gears between fisheries and this. But I guess it's not really news to anybody that the world population is growing exponentially. And the World Food and Agriculture Organization estimates that seafood consumption from 1990 to 2018 increased by over 120%. During that same time span, 1990 to 2018, capture fisheries have yielded about 14% increase in production. And this is all while aquaculture, sneaky, has been increasing by over 570% during that same time span. So NOAA estimates that here in the United States, we import over 90% of the seafood that we consume. For me, this leads me to believe that we could be producing more seafood from aquaculture and joining the rest of the world with a potentially very ethical and sustainable form of seafood production. So I've been focusing my research on tautog, which is a local fish species native to these waters. Their range extends from Nova Scotia down to South Carolina. However, the highest abundances are Cape Cod to about the Delaware Capes. What's most interesting about tautog when comparing them to other aquaculture species is that they eat primarily invertebrates. And this is different from fish that require more fish to be fed because, you know, catching wild fish to feed your farmed fish doesn't really mean it's a very sustainable seafood solution. So tautog eat green crabs in this area, and this is an invasive species to New England and the Pacific coast as well. So our lab has been able to pay fishermen to catch green crabs, and we're feeding them to our tautog and comparing them, the growth rates between tautog fed green crabs and tautog that are fed commercial fish pellets. Preliminary data shows that tautog growing off of green crab are growing 2 to 3 times as fast, which is amazing. And in the future, I'm looking at a funding project that just got through the Sustainable Agriculture and Research Education Group based out of Vermont to be looking at behavior and feeding frequency with tautog in aquaculture. In the process of acquiring another grant right now as well with partners down in Sandy Hook, New Jersey, to continue our tautog research, that time at the larval scale. So it's my belief that tautog could be an entire industry that we could make here in the United States and really uplift coastal communities. I hope to revolutionize the way that we talk about farm seafood. Kind of gets a bad rap between environmentalist groups. So stay tuned, follow me on Twitter, or don't if you're not a Twitter person. I just kind of became one myself recently, but reach out. I'd love to talk shop, and thanks for listening to me talk.
Thank you, Mike. Please thank Mike for a good talk. And I am going to move it to our next speaker, who is going to be another talk from Tricia Perez, this time focused on local seafood dynamics.
Hi, it's Tricia, me again. So when I first heard about this talk and that there was going to be a cooking demonstration with monkfish, I believe, it just got me excited about using local species and underutilized species. And so that kind of led me to wanting to talk a little bit about how I connected with underutilized species through a nice silver lining of the pandemic. So coronavirus has touched all of us, our families, our work life, our social life. We all stayed inside, restaurants closed, the economy suddenly stopped, and all sectors took a hit, including commercial fishing. And with the restaurants closing globally, the prices for seafood dropped and the market for seafood seemed to completely halt. And so Rhode Island has a really strong commercial fishing fleet. They have bottom trawls and gill nets, lobster boats, trap boats, occasionally a longline boat, scallop boat. And so these fishermen steam out onto the ocean for a day or up to 2 weeks to fill the boat with squid, flounders, whiting, monkfish, or a little bit of everything. And so when they come back to land, they offload their catch at a fish house. And this fish house processes the fish if needed. For example, filleting the flounder or cleaning the squid of its ink and guts. And then the fish house sends the fish to market. So what does a fisherman do when the fish house won't take your fish? Well, what if they could just sell it directly to us, the consumer? So in Rhode Island, fishermen have been able to sell their lobsters or crabs directly to consumers on the dock for years, and it's been really popular down in Point Judith. But on April 17th, 2020, the state of Rhode Island passed an emergency regulation creating direct sale dealer licenses for fishermen. So this allowed the fishermen to sell finfish right to the public. So this photo is of the Curtesis family selling their fish on fishing vessel Christopher Anthony in Newport. And so they just jumped on this opportunity, and they used social media all summer to promote themselves, and they posted up at the pier in Newport Shipyard every Saturday to sell directly to the public. So of course, I was there every Saturday to get fresh fish. And so I was really excited to support my local fishermen, and it was an incredible opportunity to try cool new fish. Like we got a skate in that left photo, we got whiting, and we got scup a lot. This is grilled scup, the top right photo, $1 each for the scup. So it's a great price for me. That's a higher price than they would have gotten at the fish house. And then the silverfish is a weak fish, and they only caught one that day, and the fish house isn't going to just take one fish. So it was really cool to be able to try new things and support the fishermen. So one obstacle of this process is that they have to sell them whole. I brought whole fish back, and I watched a lot of my neighbors decide not to get fish because they had to fillet it themselves. The fishermen weren't able to fillet it due to even higher health and safety concerns due to coronavirus. So if anyone does get whole fish, it's really not hard to fillet. You can eat them whole like I did the scup. So I encourage everyone to try to connect more with their local seafood. And so while 2020 has been pretty much the worst year, I think the flexibility given to fishermen by the state of Rhode Island was one silver lining. And so there's this Fishline app that everyone can download where fishermen can log into the app and let people know in your area that they're selling directly at the dock. And I think this app works around the US. So if you travel to another coastal port, you might be able to find local fishermen wherever you are. And I encourage everyone to check out Eating with the Ecosystem, which is a really awesome nonprofit I'm involved in, promoting a place-based approach to sustainable seafood. It has plenty of tools and information on how you can support your local fishermen. So check that out. And you can ask me any questions, follow me on Twitter, new to it as well. But yeah, thank you for listening to me talk again.
Thank you, Tricia. I can echo the amazing resources on Eating with the Ecosystems website. There's some fantastic sets of recipes there, which I've used a lot this year too. So our other speaker has actually emailed me a video, which is really exciting. So I think we'll be able to still hear from Kim. But while I get that set up, I think I'm going to give a chance for people to ask recent speakers some questions. So if you have questions for Tricia about local seafood markets, questions for Mike about Tautog aquaculture, questions for Amanda and Ali about the interactions of fisheries with wind farms, please go ahead and either unmute yourself and ask them directly, or write your questions into the chat and we'll deal with them.
I just have a question for Amanda about the Jonah crabs. My family and I go up to the coast of Maine every summer, and the water up there is very cold, and the Jonah crab seems to be abundant. You see it for sale everywhere, and it's delicious. And I come home and I can't find it. It's very rare that I find any Jonah crabs in markets. Do they like the cold water?
They do, but we actually do find Jonah crabs from Florida all the way up to Canada.
Really?
So it's not specifically a New England fish, but for some reason it's just not popular in Massachusetts, but they are delicious. I have seen them. The only place I've actually seen them like out in public is at Encore in Boston. They had them at their buffet, but I've never seen them in stores either, but they are delicious.
They are. Thank you.
So I have a quick question. Well, I don't know how quick it is. It's kind of long, but so usually I hear from people who are, PhD or grad school candidate doing research in, literature or things like that. So, and they always say that like they come across something unexpected. And I wonder if any of you or all of you are far enough in your research to have found like something unexpected or like an offshoot of what you're doing that is drawing your interest and makes you want to pursue something else. And I mean, not stop your research, but like, wow, now I'm going to have to research that. And so I just wonder if any of you have come across that.
Yeah, for me in particular, I guess, I mean, it still has to do with Tautog, but I've noticed that they're really skittish, like more skittish than I initially thought. So like if you feed like over a meter away from the tank or from their shelter, they like hide all day long. They'll like sleep all day long. It's like, do you even have fish in here? They look like they're dead. And it's like, no, I have fish in there. I promise there's like 50 fish inside that little PVC pipe. But if you feed them too far away from their shelter, some of them just like, that's way too far, man. I could get killed out there. They like won't even go that far. So the next phase of my research is going to have to do with like sequential feeding and seeing like how adventurous these fish end up being. And that's just one of the things that no one really knew, or at least, I mean, no one that I'm in close proximity working with knew. So it's just one of those developments that happens. And I know there's going to be something else that comes from that. Yeah. Again, that's like what drives you mad and also keeps you going all at the same time.
I know for me in my research, in like a similar vein, so the finding of the slope sea larvae of Atlantic bluefin tuna in 2016 was highly unexpected and is still pretty contested within the scientific community in terms of the importance of the spawning ground. But it also opens up a lot more questions about if we're finding larvae unexpectedly in this location, what other locations where we might find them. So there have been some indications that there might be other spawning grounds. I'm not sure how significant, but around the Bahamas, Cuba, and the Azores and Canary Islands. So those are all avenues that are largely unexplored in terms of where spawning could be taking place. So there's a lot of unexpected avenues that, you know, this work, even just focusing on one species, could take anybody.
Yeah, wow, it's great. I want to mention that I did put the link to watch the cooking demonstration, which does include deboning, you know, it starts to finish. And the chef who is doing it, is a specialist in, a culinary specialist in cooking of ugly fish. So he likes to take some weird looking fish that people wouldn't really buy because they're ugly, and teaching them how to cook it. And I think if you Google ugly fish cooking, you can make use of some of those fish you're getting at the docks in Providence. And teach your friends who might be afraid.
That's great. We have a couple of minutes left, so I'm going to try and play Kim's video through Zoom. And I know sometimes that that's not the best way of viewing video, so we'll try it and see if it works. Kim's down in North Carolina doing field work in terms of doing some fishing, and so she was able to sort of scramble to find time away from the fishing to do this, but it doesn't seem like her connection is working enough. Can you see this video? Yes.
Yes.
Okay.
Yeah.
Oh, no. All right.
Here in Supply, North Carolina,
I can't.
with South Atlantic black sea bass pot fishers on.
Can people hear that?
Yeah.
Yeah.
Developing ropeless fishing technologies for their industry. For the last 10 years, they've been experiencing some real difficulties in this pot fishery as they adhere to regulations from our U.S. government that ask them to move their fishing effort offshore between 30 and 35 miles away from where they would normally fish. This is to reduce the threat or potential of entanglements of North Atlantic right whales, which are critically endangered right now. They number only about 400 in the world. So these are some of the gears here that we've been working with the fishers here to see which of these different technologies might be able to help them potentially access this really important and vital economic fishery for them closer to shore, which makes it more financially feasible and really just better overall. There's only about 30 black sea bass pot fishers down here in the South Atlantic that still have endorsements for this type of fish. So they're a really underserved community of fishers, and that's one of the main reasons why I've targeted them as a group to work with. They've been really seeing this technology as offering them a new opportunity. They've had a lot of really great experiences with not only North Atlantic right whales, but humpbacks. They're really focused on not just conservation of whales and other cetaceans, sea turtles, but also the fish that they really depend on to feed their families and to pay their bills. So we've been down here several times over the last 2 years. We did a lot of work with them earlier in May down in Georgia. We did a lot of demonstrations, dockside demonstrations for fishers, as well as members of NOAA, different conservation groups. And I'm here right now and have been here since the beginning of September with an amazing group of volunteers that actually started out from a whale conservation side of things. So they've been going out with me and these different fishers almost daily, fishing alongside of them, working all this gear, hauling pots, doing everything that a fisher would do in his daily life. And we're really building some awesome relationships between both conservation and the fishing industry, sort of just having everybody get to know each other as people on an individual level, strengthening those bonds so that they can really work well together with these technologies going forward. We're really lucky to have all of this gear donated to us for this project by all the manufacturers that you see represented here. Anyway, so it's been really great, and it's just another step towards having the most sustainable fisheries that we can have here in the US. Thanks.
Great. Well, that worked out very nicely.
Yes, it did.
I guess Kim, she's doing work related to technological workarounds, and I think that was evident right now. So,
yeah, very good job.
Right. So I wanted to make sure that we have time for any other questions that people may have. I know that there is another session coming up very soon.
There is. Does anyone have another question? I have put the link to the video cooking demonstration in the chat if anyone would like to join us.
Yeah, great. I would also like to say if anybody didn't manage to scribble down people's contact information during their presentation, feel free to contact me and I can put you in touch with our students. And if you want to learn more about SMAST and the work that's happening, we had a very nice range of the work that's going on with our faculty and students today. But if you want to learn more, you can check SMAST out on the web. And my science communication class is actually taking over the SMAST Twitter account this semester. So if you go to, if you follow SMAST on Twitter, you will hear, you will read posts from many of the students that you've heard from today. And I also want to thank Anne-Marie and the rest of the homecoming weekend organizers once again for giving us this opportunity to talk today. And be sure to check out the rest of the events. Thanks very much.
No, thank you, Gavin, and thank you too to your students. This has really been illuminating. I'm sure everyone has enjoyed it tremendously. We appreciate people who have joined us for homecoming, and homecoming is not limited to alumni. It's our friends and families as well, and certainly students. So if you would like to join us on any of the remaining events, please do so. You can get there at umasd.edu/homecoming, and the links are right there now, so you can just pop in and visit anything that's going on. So thank you so much, Gavin, and thank you all of the students for telling us about your research. We appreciate it. We appreciate your time on a Saturday.
Thank you for being here.